Lecture 49 of BMS 300. Today we will be wrapping up with the digestive system, and then we will briefly start with the urinary system. Right, the goal is to finish the urinary system by Friday, and whatever we complete on Friday will be on exam 4. Have you all had a chance to pick up your exams from the office, from the TA's office? Okay, if not, please do that, because the final exam will be quite similar to the questions you had on the first 4 exams. I would highly recommend that you understand how those questions were worded. Yes? Yes. Ask Michelle right there. So contact the TAs and they can give it to you. So with that, let's look at an overview of what I hope to cover today. So we will be talking about fat metabolism. We'll see how fats are broken down and how they will be eventually absorbed into the digestive tract. And as we see the transport of fat taking place to various organs, we see that there can be some abnormalities which happen, right? And if something goes wrong, it can lead to this condition known as atherosclerosis. So that's the pathology we'll consider in terms of fat metabolism. So we've talked about atherosclerosis briefly before, where you will have the blockage of blood flow because of this plaque development. I mentioned this when we discussed about heart attack in the cardiovascular system. But what we'll do now is we'll get into the mechanistic details of how the plaque actually develops and what are some ways we can prevent this plaque from developing. And then we'll move on to protein metabolism and see what are the various steps involved in protein breakdown, and we'll consider the various organs involved in this and what types of secretions are released by various organs for protein metabolism. And then one pathology which we'll consider in terms of protein metabolism is kwashiorkor. So this is a condition caused by protein deficiency, and in this picture here's a child affected by kwashiorkor, and we'll talk about some of the clinical manifestations of kwashiorkor and what is the underlying mechanism as well. So when we think about lipid metabolism, let's say you eat some food which is high in fat, like a juicy burger, it's going to start right in the mouth. The lipid metabolism will start in the mouth because there will be the lingual lipase, or also known as salivary lipase, which is going to break down the triglycerides into the fatty acids. And this fat digestion, which begins in the mouth, is going to continue in the stomach because of gastric lipase as well. And I hope you all understand why we need a salivary lipase versus a gastric lipase, because the gastric environment is very acidic, and so you need a lipase which can work in an acidic environment. So the breakdown of fats in the mouth and in the stomach is not the majority of the fat breakdown. The majority will take place in the small intestines. And so when the chyme is exiting the stomach and as it enters the duodenum, the fats are detected by cells in the duodenum which will release CCK, also known as cholecystokinin. And CCK has 4 major functions, and we'll see 3 of these functions play a role in fat metabolism. Firstly, CCK is what acts on the liver and triggers it to produce the bile, right? And the bile will be stored in the gallbladder, and CCK will act on the gallbladder, cause it to constrict and squeeze the bile into the duodenum. Thirdly, CCK will also act on the pancreas and cause it to release a number of enzymes, and some of these enzymes are used for breaking down fats, so they are the pancreatic lipase. So we see that majority of the fat breakdown will take place in the small intestine. So now let's see what bile does, right? So bile is going to help with the emulsification of triglycerides. And what we mean by emulsification is that it's going to make these large fat globules into these smaller droplets of fat. And in order to do this, we need to understand the structure of bile. So if you look at the bile acid, it's got two parts. It's got a hydrophobic region and a hydrophilic region. So as an amphibolic molecule, we see that this hydrophobic part will interact with the fat, whereas the hydrophilic region will be exposed to the aqueous environment. And this is quite similar to what we discussed, I believe, in lecture 2, where we talk about soap emulsifying grease, and the hydrophobic region will interact with the grease particles and the hydrophilic region is exposed to the aqueous environment. Now the main purpose of this emulsification is so that there will be these micelles which are formed. Micelles are referring to the soluble complex of the bile acids with the fats, and these micelles, they will help in the fatty acids moving into the small intestine cells. So the intestinal cells will have the fatty acids moving into them, and the reason this is possible is because the micelles will have a larger surface area to volume ratio when you have these smaller droplets, and so the pancreatic lipases can more effectively break down the fats, and as the triglycerides are broken down into free fatty acids, they can enter into the intestinal cells. Is that clear why micelles are important? So these free fatty acids, once they enter the intestinal cells, they are going to be rebuilt into triglycerides, which will then be put into these chylomicrons. So chylomicrons are a type of lipoproteins, and lipoproteins' main function is to transport fat from one part of the body to the other. Now lipoproteins have two parts. They have this inner core, which is made up of triglycerides and cholesterol, and this outer shell, which is made up of phospholipids with proteins embedded in the phospholipid layer, and it is because of these proteins that the lipoproteins have their name. Now the proteins in this shell, they are going to have a unique signature which will act as the address on an envelope, and it can specify what is the destination for these lipoproteins, right? And we will not talk about the unique protein signatures between the various lipoproteins because that is beyond the scope of our discussion. But anyway, these chylomicrons, they will carry the fat through the lymphatic circulation, and they take it all the way to the liver. And in the liver, these chylomicrons will be repackaged into a different type of lipoprotein called VLDL, which stands for very low density lipoprotein. So now let's take a look at what are the various lipoproteins which are involved in the fat transport. So in this figure here, notice that chylomicrons, they are really large compared to the other types of lipoproteins, and we see that there is VLDL, IDL, LDL, HDL, and I will tell you what is the meaning of each of these abbreviations, and also what is the purpose of these various lipoproteins. Do you need to know the sizes mentioned here? No, you don't. I'm just telling you so that you have an approximate idea of how their size differs. So chylomicrons, they will carry the maximum amount of fat just because they are much larger, and they will carry fat from the epithelial cells to the liver by the lymphatic circulation. And the VLDL, they will be produced in the liver, and they are going to transport fats to muscle and adipose tissue. And when the VLDL goes to the adipose and muscle tissue and it gives up its fat, it will turn into an IDL, which stands for intermediate density lipoprotein. So as the density of these lipoproteins is decreasing, we see that it will have a higher amount of fat, but as its density increases, it will have lower amount of fat. So this IDL can in turn go to other tissues, and it can lose more fat inside it, and it will turn into a LDL or a low density lipoprotein. Now keep in mind that all of these lipoproteins from chylomicron to LDL, they're all classified as bad cholesterol, whereas HDL, which stands for high density lipoprotein, is called the good cholesterol. And the reason for this is HDL is involved in reverse cholesterol transport. So if there's LDL particles in various parts of the body, HDL will help to move that LDL back to the liver, right? And I'll tell you why this LDL can trigger atherosclerosis. So the main question you may be having is, how are these lipoproteins losing the fat inside them, right? And for this to happen, there is an enzyme known as lipoprotein lipase. This is found in the various tissues to which the lipoproteins carry the fat to. So it's found in the capillaries of the adipose tissue, liver, and in the muscle tissue as well. And this lipoprotein lipase will stimulate lipolysis, which means it will break down the triglycerides into fatty acids and glycerol. And the fatty acids can enter into the cells where they can be used for making ATP, or they can be stored as fat. So these are the various players involved in fat transport, and next we're going to consider how they actually play a role in this, right? We've seen what are their functions, what are their names, how do they actually help in fat transport. And so for your reference, I have a insert of the various lipoproteins, and let's see how they play a role in fat transport. So the chylomicrons, they will carry fat to the liver, and in the liver we will see that they will be broken down and reassembled into VLDL, which will then be carried to various cells and tissues. And in the cells and tissues, the fat will be broken down by lipoprotein lipase, and as a result these lipoproteins will get smaller, and VLDL can be converted into an IDL, an intermediate density lipoprotein. Now this IDL has two options. It can either go back to the liver where it is packaged into another VLDL molecule, or it can go to other tissues where it can lose more fat and it can be packaged into a LDL. Now LDL also has the same two options. It can either go back to the liver and it can be made into VLDL, or it could end up in an injured artery where it can trigger the very first step of this atherosclerotic plaque formation. Okay, so in order to understand how this LDL can eventually lead to plaque formation, we need to take a closer look at the structure of a blood vessel. Yes?
So is IDL just like the storage? Or like what's its overall function?
So the difference between IDL, LDL and VLDL, for the sake of our discussion, all I'm saying is that they differ in their size, right? But another major difference is the type of proteins found on their surface. So the protein signature is going to be unique, and that will indicate their destinations, right? But the key is all these things here will help in fat transport. Okay, so let's take a closer look at the structure of the blood vessel. So we see there are three layers to a blood vessel. The innermost layer is called tunica intima, and this tunica intima has got two parts. It's got this layer of endothelial cells, the endothelium, which is directly in contact with the blood, right? And this is supported by a layer of connective tissue shown here in light purple. I haven't labeled it in this figure, but a lot of action goes on in that connective tissue, as we'll see in the next few minutes. Now the layer surrounding the tunica intima is the tunica media. This is made up of smooth muscles, and the outermost layer is the tunica externa, right? And what I'm going to do here is I'm going to give you a few details from a recent article which talks about the steps involved in atherosclerosis, and what I'm showing you here is a redacted image, right? I've cut out a lot of details which I think is beyond the scope of our discussion, but if you are curious and interested, I would request you to look at this article, right? And I do think you have enough background in terms of the immune system to understand what's going on if you were to actually go to this article and take a look. So anyway, so there are three panels I want to walk you through to say how this plaque is being formed. So first and foremost, notice that this plaque formation generally takes place at a point where the blood vessels will bifurcate, right, where they're going to branch. And the reason for this is the point where the blood vessels branch, that's where you'll have turbulent flow, right, and because of this increased turbulent flow, you have high fluid shear stress experienced at the points where there's bifurcation. And also because of the turbulent flow, the blood cells can also bounce off the branching blood vessels, and it can lead to tissue damage. But bottom line, notice that because of this turbulent flow, LDL particles shown here in gray, they can get in between these endothelial cells, right? And when they get into these endothelial cells, they can get oxidized, and when they get oxidized, notice that they are shown in red color. So when they are unoxidized, they are shown in gray. Once they get oxidized, they are shown in red. And these oxidized LDL particles can interact with the collagen, which is in the connective tissue underlying the endothelial cells. The oxidized LDL can also do another thing. They can cause activation of the endothelial cells. And what we mean by this activation is that it starts producing receptors to which white blood cells can bind. And if you remember back to what we discussed in the innate immune response, when there's bacterial infection because of a splinter getting through your skin, I told you that these selectin receptors will be expressed on the blood vessels, and that's why macrophages can bind to them, or neutrophils can bind and enter into that tissue. So similarly, this monocyte would have minded its own business and gone right along, but because of these receptors, it binds to it, and it can squeeze through by a process known as diapedesis, and lo and behold, this monocyte is now in the connective tissue underlying the endothelial cells. And this monocyte can differentiate to form a macrophage, and this macrophage can start phagocytosing the oxidized LDL particles, and it will turn into a foam cell, right? So the bottom line of this figure is that oxidized LDLs are phagocytosed by macrophages to form foam cells. So foam cells, they are microscopic, but as they start aggregating, they can lead to what is known as fatty streaks, which can be visible even with the naked eye. So there's another thing which is happening during this process. Notice that in the tunica media, some of the smooth muscle cells can migrate out of the tunica media, and they can differentiate into macrophages, which can also start gobbling up the LDL, and it forms more foam cells. So these are smooth muscle cell derived foam cells. A second thing which these smooth muscle cells do is they can lead to a formation of a fibrous cap at the place where this artery was injured, and this fibrous cap has a protective function, right? Notice that it's made up mainly of the connective tissue and the smooth muscle cells, and it's going to prevent this plaque from rupturing when you have this fibrous cap formed. But in the meanwhile, the foam cells which were produced in the previous steps, they will start dying, and they will form a necrotic core. So you can have more and more of this foam cells dying, you have cholesterol accumulation, and so this plaque starts getting bigger. Notice there's also some calcification taking place here, and the reason for calcification is some of these smooth muscle cells can differentiate into bone cells, and that's why calcium starts getting accumulating in this region as well, right? And so these are the details behind how the plaque starts developing. And now let's zoom back to a bird's eye view of what is taking place, right? So notice that the LDL goes to an injured artery, right, and there it gets oxidized, and once it is oxidized, it can signal the immune system. And what happens here, we discussed in the previous slide, that the receptors are being expressed, monocytes come in through diapedesis, they turn into macrophages, and eventually as there is more foam cell accumulation, it leads to fatty streaks being formed. And we find that there's also calcium accumulation because some of the smooth muscle cells have differentiated into bone cells. And we see that the overall diameter of the blood vessel is going to start decreasing. And you know that the capillaries, they have only a slightly larger diameter than the red blood cells, and so if you decrease the diameter, we can have red blood cells starting to build up in these blood vessels, and blood flow can be blocked. In some cases, the plaque can also rupture, right? So what causes the plaque to rupture is the nature of the plaque. So if it's got less fat accumulation, then the plaque is quite stable. But when you have a larger necrotic core and more fat starts accumulating, it destabilizes the plaque and it can eventually rupture. And once it ruptures, we see that a blood clot can start forming, and the blood clot can eventually block the blood flow, and that can lead to thrombosis. So thrombosis can, if it happens in the blood vessels in the brain, that can lead to stroke. If it happens in the heart, that can lead to a heart attack. And notice that the stroke and heart attack does not need to take place only after thrombosis. Just decreasing the vessel diameter is sufficient to lead to heart attack and stroke as well. Yes?
What's the relationship between calcium and the foam cell?
So it can cause the plaque to get hardened. Yeah. Hardening of the plaque. Any other questions? So in this entire series of steps leading to atherosclerosis, right, all of this could have been prevented if you had a high enough level of HDLs, right? And I told you HDL is involved in reverse cholesterol transport. HDL will go around the body looking for LDL particles, and it can take the fat inside the LDL back to the liver. So we're going to use this same figure shown here to see what are some of the ways in which we can treat atherosclerosis and prevent its progression, right? So the various interventions can happen at different levels of disease progression. So the first thing is, how can we increase the HDL levels, right? There's no easy way to do it, you know. If you exercise, that can increase the HDL levels. Another way is you could have small amounts of alcohol, right? Maybe around 1 ounce per day. But the hard part with the second thing is moderation is key, right? If you exceed the 1 ounce limit, it can lead to other complications like cancer, liver damage. So highly recommend exercise. We also see that there can be some medication which is taken. So statins, they're a type of drug which will block cholesterol synthesis, and it can act on the liver to increase its sensitivity to LDL. So if LDL has there's higher sensitivity for the LDL particles, they can be taken up by the liver and repackaged into VLDL. Right, and the thing with statins is it has got side effects, just like how any drug in the market has side effects. And so if you want to use a more natural approach, one suggestion would be to take lots of fruits and vegetables. There was a study known as on a diet called the DASH diet for decreasing hypertension. So after 3 weeks of being on this DASH diet with high fruits and vegetables, after 3 weeks there was no significant difference between the effects they saw in those who were on statins versus those who were on this diet with fruits and vegetables. Also, increasing the intake of unsaturated fats. So unsaturated fats come mainly from plant sources. Examples are like olive oil, canola oil, whereas saturated fats will be from an animal source. And what unsaturated fats do is they increase the sensitivity of the liver for both LDL and IDL receptors, right? So the IDL and LDL can be taken up easily by the liver when you have higher unsaturated fats in the diet. Things you can reduce could be the overall calories, because whether the calories come from proteins, carbohydrates, or fats, they will all eventually be turned into VLDL, which can increase the LDL levels, but decreasing the overall calories can reduce the LDL levels in circulation. Also, trying to reduce the stress as much as possible, because when you have lower stress, you will eventually have lower VLDL. And I want to tie this back to slide 27 from part 3. So if you don't know what I'm referring to, this is the slide, right? So we looked at how when there is low blood glucose level, how the body responds to that. And so one example of when you have low blood glucose level is, let's say you haven't been eating for a while, and then you have the sympathetic system being activated, you're hangry, epinephrine is released, and that can activate the hormone sensitive lipase, which is found in the adipose tissue. And hormone sensitive lipase will break down the triglycerides in the adipose tissue and release free fatty acids, which can go to the muscles and be used for making ATP. So when you are stressed out, you will eventually increase the VLDL levels. But at that time, if you, let's say, go exercise, and you use those VLDL particles for making ATP, you're all good. But if you just sit in the same place, you will eventually increase the LDL levels, which can promote the progression of atherosclerosis. So when stressed, go for a run, right? Highly recommend exercise. Also, lowering the levels of nicotine is also very important, because nicotine just does the opposite of what you want it to do as far as fat metabolism is concerned. Not only does it decrease the HDL levels, but it also increases LDL levels as well. So two other interventions which you can engage in is either taking a baby aspirin or having the appropriate omega-3 to omega-6 fatty acid ratio. So omega-6 fatty acids, they are very prevalent in the American diet because of the ubiquitous use of corn pretty much in everything we eat, right? Whereas omega-3 fatty acids, they are in foods like fish, and omega-3 fatty acids will help to reduce inflammation, and in general it's just referring to the overall immune response, which can lead to foam cells. It can also decrease the blood clotting, and it can cause dilation of blood vessels. So having this appropriate omega-3 to omega-6 ratio can affect both of these steps in the progression of atherosclerosis. Is this clear, folks, how the various interventions, how they tie into the progression of atherosclerosis and how they can help to treat it? Okay, so with that, we move on to protein metabolism. Right. So we find that protein metabolism, the chemical aspect of it does not start in the mouth, right? There's no proteases released in the saliva. So the mechanical digestion of breaking down the meat into smaller particles, yes, that takes place in the mouth. But it is in the stomach where you have the hydrochloric acid unfolding the proteins and thereby increasing the surface area available for pepsin to break it down into amino acids. And as the amino acids and the undigested proteins move into the duodenum, CCK will detect the presence of proteins, and that can act on the pancreas, which will release pancreatic proteases, which further digest the proteins into amino acids. And these amino acids can be absorbed into the villi of the small intestines, and that enters the bloodstream. But keep in mind that the amino group in amino acids cannot be used by our body for making ATP, so that has to be stripped off by a process known as deamination, and that will be excreted as urea. And we'll be talking about how urea is excreted in the urinary system, which we will cover in the next couple of days. So whenever there's a excess balance of calories, right, you have too much calories compared to the amount of caloric output, we find that it will be converted into fat and stored as fat. So with that, let's consider how we can have the adequate protein quality in our diet. So it's been said that meat has one of the highest biological values for humans in terms of dietary protein, and the reason is meat is said to be a complete protein because it has all of the essential amino acids which are body needs in the right proportions. Whereas if you look at plant sources of proteins, it is limiting in either one or more amino acid, right? So that's why plant proteins are said to be incomplete proteins. And so if you take two foods, right, two plant-based foods, which is maybe lacking, one is lacking in lysine, the other one is lacking in methionine, and you mix them together, you get a complete protein, right? So examples of complete proteins include legumes and grains. So this could be beans and rice, or peanut butter and bread. So bread would be the grain, peanut butter is the legume. Tabbouleh grain and bean salad is also a way of getting complete protein. So there are ways to get complete proteins without eating, adding meat in the diet. But if you are like me, and if you prefer to have at least a little bit of meat in the diet, I actually prefer more, you could add a complete protein source to an incomplete protein, and that will also result in complete proteins, right? So examples of this include macaroni and cheese, cereal and milk, or even meat stir fry with chicken or the meat of your choice. So now let's talk about having a lack of dietary protein, right? So we see that there's this condition called kwashiorkor, and at first glance this child may look quite healthy, right, and chubby, but we see that this is caused by a diet with low proteins, but there is sufficient calories, right? So this is different than marasmus, where you just have insufficient calories. So marasmus is where you have insufficient calories, but kwashiorkor is where you may have sufficient calories, but there's a lack of protein in the diet. So a situation where this could happen is, let's say in a developing country, a child is initially getting mother's milk and is developing normally because mother's milk is a source of complete protein. But let's say after a year or two, the mother is pregnant again, and it's the newborn who's getting all of the mother's milk, right? And so this first child, to keep him or her from crying, they may just give sugar water to satisfy the hunger, and as a result, the child can develop kwashiorkor. So let's talk about some of the characteristics of kwashiorkor. We see that one of the main things observed is there will be impaired growth, because in order for growth to take place, you need to have anabolic reactions for having protein synthesis, so that the muscles can develop, the bones can form, so that fat can also be produced. So there is an impaired growth observed. We also see that there could be high rates of infection, and in order to fight infection, the body has to produce antibodies, and antibody synthesis will be affected if you don't have sufficient proteins, and that's why children affected with kwashiorkor cannot fight infections effectively. As far as the pot belly is concerned, it could be due to edema. I'll explain the mechanism in a bit why edema is caused, but it's mainly due to worm infestation because of poor hygienic conditions, and that's why both in marasmus and in kwashiorkor you could observe a pot belly in these children. And so one of the downsides of having this worm infestation is, even if the child were given nutritious food, the worms will be absorbing the nutrition instead. So whenever an intervention is done in such cases, they would administer a strong deworming agent so that the nutrients can be absorbed by the child and not by the worms. Also, a fatty liver is observed in kwashiorkor, and the reason is, if you don't have sufficient proteins to make VLDL, fat transport is impacted, and so fat will start getting accumulated in the liver. And there can also be an impaired motor and cognitive development. So if you are constantly hungry, you have the sympathetic system being activated, and you're always in survival mode, and so you are not able to focus on fine motor skills or cognitive development. Now, the one thing to consider is that this cognitive impairment is not permanent. If these children are rescued from their situation and given nutritious food, cognitive development can take place normally, right? It's not a permanent damage which is caused. And finally, we see that there's edema where there could be fluid accumulation in the extremities of the body, like in the hands or in the legs. And so the reason for this is there is a reduced osmotic pressure due to low serum proteins. When we talked about the composition of blood, I said that blood plasma has got proteins in it like albumin and other types of proteins which bind to hydrophobic substances. And so let me explain why low osmotic pressure can lead to edema. So here's a cartoon of arterial blood shown here in the red box, and then the surrounding tissue is indicated by the yellow lines. And so in the arterial blood you will expect a lot of serum proteins like albumin. There will also be hemoglobin inside the red blood cells. And notice that you have the additional proteins also in the surrounding tissues. And there will be the water which is inside the blood. As the blood is being pumped out, due to the blood pressure, we see that some of the water will move into the surrounding tissues. And this water which moves in the surrounding tissues is going to be drawn back into the blood vessel due to the osmotic pressure which is expressed by the proteins in the blood plasma. So we see that the water can be taken back into the blood vessel. But in the case of kwashiorkor, let's say you decrease the amount of proteins in the blood plasma, then the osmotic pressure to draw back the water in is going to be reduced, and so more water remains in the surrounding tissue, which eventually leads to swelling, and that's how edema takes place. So just to summarize, we see that the blood pressure is equal to the osmotic effect due to the presence of the plasma proteins. You will not have any swelling. But if the blood pressure is greater than the osmotic effect due to the plasma proteins, it can lead to edema. So with that, let me quickly summarize what we discussed in part 4 of the digestive system. So we talked about the various steps involved in fat metabolism, and we considered the different types of lipoproteins and how LDL particles, if they get accumulated in an injured artery, how that can lead to atherosclerosis. And we talked about the various steps involved in the progression of atherosclerosis and how it can be prevented by different dietary interventions, lifestyle changes, or even through some medication. As far as protein metabolism is concerned, we talked about how it starts in the mouth as far as mechanical digestion is concerned, but the chemical digestion starts in the stomach and it continues in the small intestines. And we considered how kwashiorkor can be caused when you have lack of proteins in the diet. So with that, we have 12 more minutes, and I want to use this time to talk about the urinary system, right? So